So, good morning, class. Welcome to another session. So, we will start with the fundamentals and applications of fluorescence activated cell sorting, that is FACS analysis. So, regarding that, this is basically to sort complex and simple cells, bigger and smaller cells, and according to the morphology, how the cells have, like red blood cells, all the immune cells, or you want to check between a disease state patient and normal patient, that what are the cells configuration is. So in order to find that, the your FACS analysis is done. So it has many applications. It has been very varied, valuable in the research also, and also in the daily application from biochemistry perspective. So indeed, one of the best methods till now. So we will cover in this topic with the basics of flow cytometry, how your flow cytometry sorter works, understand how cells are sorted, and description of different cell sorting possibilities, tips on sample preparations, overview of sorting services provided by the company, this FCS. So which techniques are appropriate for my cell separations? There are two types. One is for tissue, one for fluid particles and suspensions. For the tissues, you can go for laser microdissection microscopy. For the fluids, you can go for FACS, that is your fluorescence activated cell sorting, microfluidics, or large particles sorting. So this one is the best. This we will discuss FACS. So basically, what is flow cytometry? Flow cytometry is a like flow means fluid, cyto means cells, and metry means measurement. So it's a fluid cells measurement, like all the fluids that are in cells, and we need to measure them. That's the flow cytometry. So what types of particles can we analyze? We can analyze various cells, your RBCs, WBCs, various kinds of chromosomes, bacteria, yeast, microparticles. Everything is possible in this. So what particle size can analyze? This was the like what sort of particles could be analyzed and what kind of sizes could be analyzed. Sizes could be analyzed from zero point 0.5 microbacteria to the 14 micrometer of your monocytes. Yeah. So varied sizes. So three key parameters are measured. That is relative size, relative internal complexity, relative fluorescence intensity. So all these things are matters in this. Then where are the sample runs? So samples could be run over these analyzers. These are the various machines, uh, various things, how they work. They are differentiated based on the complexity, how the results are shown, and how fast it is. So this is the cheapest one. This one we can see here, but the results are also slow then afterwards. So we can divide this system of flow cytometry into three parts, that is fluidics, optics, and electronics. The fluidics is the one to which get focus on the cell interrogation and create a stable breakup for sorting. Then optics to collect the light signal, and electronics is to propose those electrical signal into digital signals so we can communicate with the computer. So how we record these individual cells? The sample is first introduced into the running sheet fluid in the flu cell. So you have your samples, yeah, all the central core samples are there, and sheet fluid and samples do not mix. Reduction of cross section causes an acceleration of central core that leads to laminar fluidex. And sample flow adjustable. So you can see like this, they become single flow afterwards, and the sheet fluid is going on like so. Then there are two types of different optic systems in sorter. That is lasers, fiber optic cables that carry beams to steering prisms, which then direct laser beams to the stream. So here we can see that um, various beams are being used, and through the prisms they go, and they are in the nozzles go straight and give a very nice fine laser beams at them. So there are two types of optics. One is collecting the optic consisting of fiber optics that direct the emitted light to the appropriate emission, and the filters that directs the light signal in the emission block to the appropriate photon multiplier to PMT. Like so. So we have individual collection fibers. Now filters are arranged in different arrangements like A, B, C, and A, B, C like this. And photon multiplier to PMT. So you have photon of light here with the cathode and focusing electrodes and anode on another side. So they start electrons moving from here to another to there to there to there to there to there like this. So you have at the end current coming out of it. How are cells actually sorted? So samples generate light, scatters fluorescent signals. Signals are analyzed here. Then charge applied by the stream changing wires. Then these charge droplets break off into three different parts. So deflection, there are some north and south pole like of magnetic plates are there. So according to that, they get differentiated. Red go to the left, green go to the right, and in the center ones, there's the uncharged drop particles which go to the waste. So in this way, you can differentiate between your red and yellow particles. So that's how the basic principle of your cell sorting. This is how the image looks like. Your droplets is coming, and they started to go afterwards single drops, and yeah, we can have our results later on. So what other collection devices we can sort for my samples? So you can done with the purpose of enrichment of cell culture, DNA, RNA analysis, and sort of fluorescent proteins. So cell tubes that can be sorted are 1.5 ml per tubes, 5 ml FACS tubes. So various possible things are possible. And this is how the FACS from inside, the X-ray, we can say, looks like. So you have these magnetic, magnetic um, tablets are there. You have your cell sorter here on the top, and the things goes down, and this is how the things are sorted out later. So then the plate sorts. So plates could be also of 96 well, 384 well plate, 96 well plate, 8 well plate, according to the results that you're sorting. So 8 well plate, now 1, 2, 3, 4, 5, 6, 12 well plate, 1, 2, 3, 4, 5, 6, 7, 8, 9, 18, 18, 12, 8, like this. So some quick tips while you're preparing for sample preparation. So plan experiment well. Choose fluorescent antibodies in agreement with the optical configurations of the intended sorter. Note that different sorters may slightly vary in the optical configuration. So unstained non-transfected cells to estimate background signals. Bring signal stained samples to set compensation, and bring FMOS if needed to get sets, and use maximum 2% FCS BCA, which is recommended in the suspension sorted, because otherwise cells become sticky. Avoid phenol red media due to autofluorescence. Most cell types will do well in just PBS, FCS, BCA. So use calcium magnesium free sorting buffer, or add EDTA to that. So if the cell's preparation induces increased cell lysis, use 25 microgram per ml DNase, and use 1% acutase in sorting buffer. So all samples need to be filtered directly before sorting. So you can use 70 micrometer nozzle, 85 micrometer nozzle, 100 micrometer nozzles in order to clean your cells. So in summary, what we have done is that we have understood how the analyzers sort the things with the sorter. We can sort between smallest to largest bacteria, and how the fluorochrome panel is working in order to configure the optical configurations. Prepare your samples following your guidelines. Do not forget to use controls, and bring appropriate collection media for downstream technique. Thank you very much. That was the basics of your fluorescence techniques, how we do them. Uh, Now, so these videos for 10 minutes for two videos will explain the basic principle that I have also explained, but it will be more better that how the pictogram histograms are made. So let's start.
Hello guys, in this video I'm going to speak about a very important or very interesting mechanism called the flow cytometer and FACS. Actually, I did two videos, and in this video I'm going to mention specifically flow cytometer, and in the next video I'm going to mention the FACS mechanism, fluorescence activated cell sorting. Let's start speaking about flow cytometer. Flow cytometer is a cell counting mechanism. So it's used to count the cells and to analyze the phenotype and the health situation of the cell. How does it work in general? First, in general, in flow cytometer, I have something called the flow cell. In the flow cell, we add our cell mixture. Then I have the sheath fluids. The sheath fluids will apply something called the hydrodynamic focusing. The hydrodynamic focusing of the sheath fluids will send the cells one by one through the flow cytometer tube. It's very important to to pass the cells one by one through the tube, because here I have something called the laser beam, and the cells should pass through the laser beam one by one. This point where the cells pass through the laser beam is called the interrogation point. Now, when the cell passes through the laser beam, there are two events happening. First is the forward scattering, which is the light scattered in an acute angle to the forward direction, and the side scattering, which is the light scattered in every other direction, in side directions. These two events, or this light scattering, is detected by specific detectors called the forward detector and the side detectors. And these detectors are connected to a PC, or a data analyzer, which is going to analyze the data obtained from the detectors. Now, this is in general. Now I'm going to speak more in details. Saying that this is the flow cytometer tube, and then the cells pass one by one through the laser beam. When the cell pass through the laser beam, the cells scatter the light in a forward direction, which is the light scattered in an acute angle. This forward scattered light is directly proportional to the size of the cell. Why? Because the cells scatter as like a particular amount of light in the forward direction, but when the cell is bigger, the forward scattered light will be larger, or like more forward scattered light will be detected. And when the cell is smaller, the cell will scatter less light in the forward direction. So the forward scattered light is directly proportional to the size of the cell. Now the detector detects the pulse of photons, which are the forward scattered light is a pulse of photons, which are detected by the detector. And the detector then is, as I told you before, is connected to a PC, or a computer. The PC will convert the intensity of the light detected by the detector into voltage. And then I will get a specific amount of voltage for every cell passing through the laser beam. The PC will give me a data similar to this. So every time a cell passes through the laser beam, I will get a specific peak, like high voltage peak for large cells and low voltage peaks for small cells. The data, or the forward scattered light data at the end, will be similar to this. Time versus voltage, and then the peak will be proportional to the size of the cell. This is forward scattered light. The second event happening in the flow cytometer is the side scattered light. So when a cell passes through the laser beam, the cell will scatter the light in side direction. And this side scattered light is directly proportional to the complexity and granularity of the cell. Why? Because the simple cells scatter the light in the side direction, yes, but when we have granules inside the cell, so when the cell is complex from inside, these little granules will also scatter light in different directions, and in this case I will get more side scattered light, like this. So this is a complex cell from inside. It contains small granules inside, and then these granules will also scatter the light in different directions, and in this case I will get higher intensity of the side scattered light. Similarly to what we saw in forward scattered light, the detectors are connected to the PC, which is going to convert the intensity of light into voltage, and then I will get something similar to this. So the side scattered light data will be similar to the forward scattered light data. So for the complex cells will give me a high peak, and the simple cells will give me a small peak. Now, in order to analyze the cells, what I have to do is that I can combine both forward scattered light and side scattered light data together. In in this case, I'll get something called the two-dimensional histogram. The two-dimensional histogram, from its name, it's two-dimensional. So I have side scatter and forward scatter data all together. In this case, what I get is something similar to this. So I have here 4 quadrants. If the cell appears in the lower quadrants, it means that it has low signal on the side scatter detectors, which means that the cell is simple. If the cell appears in the upper half, it means that it gives high signal on the side scatter detectors, so it means that the cell is complex. If the cell appears on the right half, it means that the cell gives high signal on the forward scatter, which means that the cell is big. And if the cell appears here, it means that the cell is small. So if the cell appears, for example, in this quadrant, it means that the cell is small and it's simple. If the cell appears here, it means that the cell is small but it's complex. Here, it means that the cell is big and complex, and here, it means that the cell is big but simple. I will give you a very good example of this, which I have an example about the analysis of a sample of white blood cells. So what we see here is an analysis of white blood cells. This is side scatter and this is forward scatter light. What we see here is a group of cells which are complex, very complex cells, because they give high signal on the side scatter detector, and they are medium inside. They are not so big and they are not small, so they are medium inside. Obviously, these cells are granulocytes, because granulocytes, or like eosinophil, basophil, neutrophils, they are medium inside, but they are very complex cells. Here we have bigger cells because they give almost like they give more or less high signal on the forward scatter, but they give low signal on the side scatter. So they are big, but they are not complex. They are simple cells, and these are monocytes, because monocytes are big cells, but they are not complex. Here we have very simple cells because they give very low signal on the side scatter, and they are like smaller than monocytes and granulocytes. So obviously these are lymphocytes, because the lymphocytes are smaller in size and they are simple cells. Of course, this is a very useful technique used to count the cells and to analyze the phenotype and the health situation of the cells. And what's very special about this technique is that we can analyze many thousands of particles and cells per second. It's like it's an easy technique. It's not very hard to perform, and it's precise. It's very used nowadays in all the laboratories. This is everything I wanted to tell you about flow cytometer, which is very important to know if you want to know more about FACS mechanism. In the next video, I'll speak about FACS mechanism. So if you want to know more about FACS mechanism, then you need to watch the next video. If you like this video, please don't forget to like, share, subscribe to the channel, and if you have any questions, leave it in the comments. I will answer you, and see you in the next video. Bye.
So this was the 2D histogram. That is how it is done according to the complexity of the cells, according to various sizes. Now let's check another one.
Hello guys, this is a continuation of the subject flow cytometer and FACS mechanism. In the previous video, I spoke in details about flow cytometer. And in this video, I'm going to speak about FACS mechanism, fluorescence activated cell sorting. If you didn't watch the previous video, I highly recommend you to go and watch it, because it will be much easier to understand FACS mechanism if you fully understand the flow cytometer mechanism. So let's start speaking about FACS. FACS mechanism is not only a cell counting mechanism, but it's also a cell sorting mechanism. So it provides a method to sort a heterogeneous mixture of biological cells. It depends on light scattering, as we saw in flow cytometer, so forward scattering in light and side scattering light, but it also depends on the fluorescence, and it also can detect fluorescence. FACS mechanism looks like this. So we have the machine, we have of course the computer, which is going to analyze the data, and we have the fluids container and the waste container. Now, how does it work? A FACS machine looks like this from inside. So we have also the flow cell and the sheath fluids, which apply the hydrodynamic forces on the cell to pass them one by one through the tube. This is what we saw in flow cytometer in the previous video. The cells will pass one by one through the cell tube, and then we have the laser. So the cells will pass through the laser beam one by one. When the cell pass through the laser beam, there is the forward scatter, which is detected by the forward scatter detector, and then there is the side scatter, which is detected by the side scatter detector. But here there is also the fluorescence, which is detected by the fluorescence detectors. Here we have mirrors and filters, which are aimed to filter the light and to deliver particular wavelengths to the appropriate detector. Then all the detectors are connected to the computer, which is going to analyze the data. Now let's speak more in details. As we saw in in flow cytometer, we have forward scatter and light and side scatter. Here we have also the fluorescence. So saying that I have a cell mixture. Inside this mixture, I have a mixture of cells, or cells who are expressing a particular protein, and other cells who are not expressing the particular proteins, the particular protein or the protein of interest. What I want to do here is I want to count how many cells there is inside the mixture, and how many cells are expressing the protein and how many cells are not expressing the protein. And then I want to separate this heterogeneous mixture into two groups of cells, the cells who are expressing the protein and the cells who are not expressing the protein. To do this, first of all, I have to design a specific antibody for this protein. This antibody is specific to the protein of my interest. I'm not like I can buy this protein. There are companies who design specific antibodies for particular proteins. Then I have to label this antibody with a specific fluorophore. Now, what I have to do is that I have to add this antibody, labeled antibody, to the mixture of cells I have. When the antibody is added to the cells, this antibody is going to bind specifically to the protein of my interest, and then I will have labeled cells. So I will have the cells who are expressing the protein will be labeled with this fluorophore. Now, when the cell passes through the laser beam, this is going to happen. First, I will get the forward scattered light, which is detected by the forward scatter detector, and this is proportional to the size, as we saw in the previous. We saw this in details in the previous video. So forward scattered light is directly proportional to the size of the cell. Side scattered light is directly proportional to the complexity of the cell. And then the fluorescence will give me an information about which one of the cells is expressing the protein and which one of the cells is not expressing the protein. Now, when the cell pass through the laser beam, this is the laser beam. First of all, this is the forward scattered, which is detected by the forward scatter detector. The side scatter, which is detected by the side scatter detectors. And then when the fluorophore pass through the laser beam, the fluorophore will be excited, and then when the fluorophore is excited, the fluorophore will emit light at a particular wavelength, and then this light will be detected by the fluorophore detectors. All these detectors are connected to the computer, which is going to give me the exact data about every event of these three. Now, of course, when the cell is expressing more proteins, there will be more fluorescence coming out of this cell. At the end, I will get a data similar to this. So when the cell is expressing the protein, I will get a high peak, and then when the cell is not expressing the protein, I will get a small peak. And then the computer will give me something called the one-dimensional histogram, which is something similar to this. Here we have the cell count, or the events. We call them the events passing through the laser beam. And here we have the signal of the fluorophore. So when I have a high signal of fluorophore, then these cells are the cells who are expressing the protein of interest, and the cells who give low signal on the fluorescence detectors are the cells who are not expressing the protein. What I can do further is to combine this data with other data I have. So, for example, I can combine the data obtained from the fluorescence detectors with the data obtained from the forward scatter detectors, for example. So here I have the fluorophore, here I have the forward scatter, or here I have the fluorescence and forward scattered light. When the cell appear here, it means that these cells are small cells because they have low signal on the forward scatter, and they have low signal on the fluorescence detectors, so they are not expressing the protein. When the cell appear here, it means that the cell is not expressing the protein because it has low fluorescence, but it's a big cell because it has high signal on the forward scattered light. Then when the cell appears here, it means that it's expressing the protein, but it's a small cell. And when the cell appears on the upper right quadrant, it means that the cell is big and it's expressing the protein. So this is what we call the two-dimensional histogram. I can combine two data together, and then I get something like this. Now, what's special about FACS mechanism, or why it's called FACS? FACS means fluorescence activated cell sorting, which means that this mechanism cannot only give me a cell count, cannot give me a data about the cell count or the percentage of the cell expressing the protein and not expressing the protein, but also the FACS can separate the cell mixture into two groups of cells. How does it work? Let's see. This is the FACS mechanism. So here we have the flow cell, and here we have the cells passing one by one through the laser beam. When the cells pass one by one through the laser beam, as we saw before, it will scatter light forward and side scatter light, and also fluorescence. All these events will be detected by the detector. At the end of the tube, there is something called drop formation. In drop formation, we have a mechanism called a vibrating mechanism. This vibrating mechanism will separate my mixture into small droplets. These droplets should contain only one cell by droplet. This is very important. Then what I can do as a user is that I can adjust the settings of the program in order to chart the cells. So at the end of the tube, I have something called the electrical charging ring. The electrical charging ring is responsible to charge the cells according to my adjustment. So, for example, if I adjust the settings, for example, of the program, I can ask the computer to charge the high fluorescent cells with a positive charge and the low fluorescent cells with a negative charge. Then the electrical charging ring will charge the the cells will pass through the laser beam. The detector will detect the fluorescence, and then the computer will charge the high fluorescent cell with positive charge, for example, and the low fluorescent cells with negative charge. I can choose, actually. For example, if I have this data, this is the two-dimensional histogram, I can choose to charge the big high fluorescent cells with a positive charge, and the big low fluorescent cells with a negative charge, and the other cells with no charge. Then the neutral cells will go to the waste. These cells might be like debris. These might be like just debris. I'm not interested in them, so it depends what cells I'm interested in. So I can adjust the settings of the program, and then the electrical charging ring will charge the cells. Then I have different electrodes, like negative and positive electrodes, and then the positive cells will go to the negative electrodes, and the negative cells will go to the positive electrodes, and the neutral cells will go to the waste. In this case, I will have two groups of cells: the cells who are expressing the protein and the cells who are not expressing the protein. And this is what we call cell sorting. Of course, I can also study two proteins, not only one. And this is done by using different... of course, to study two proteins, I need two specific antibodies, because I need one specific antibody for each protein. But I need to label every antibody with a different fluorophore. Of course, I have different fluorophores, and every fluorophore emits light in a different wavelength. I need to know every fluorophore, the wavelength every fluorophore emits light in. So if I use two different fluorophores, then I can study two different proteins. For example, this fluorophore emits a green light, or a light green light, and this fluorophore emits yellow light. So they are different wavelengths. Then if this cell is emitting only has only a green fluorophore, we can know that this cell is expressing one protein, or the green protein. If the cell is emitting yellow light, then the cell is expressing only the second protein. And if the cell is emitting two lights, then or giving two signals on the two detectors, then we can say that the cell is expressing both proteins. How can we detect two different wavelengths? As I showed you before, the FACS mechanism contains different detectors, different fluorescence detectors, and here we have the filters, the light filters, which deliver a particular wavelength to a particular detector. So every detector will detect a particular wavelength, and then we can use two different fluorophores if they emit light on different wavelengths. Okay. We can also combine the data together. So I can form this two-dimensional histogram again, but with protein 1 and protein 2. So I have fluorophore 1 signal coming from fluorophore 1 and fluorophore 2. So when the cells are in this, when the cells are here, it means that these cells are expressing both proteins, because they are giving high signal on the first detector on the second detector. If the cells are here, it means that they are expressing protein 2, but they are not expressing protein 1. These cells are expressing protein 1 because they have high signal on the detector of fluorophore 1, but they are not expressing protein 2 because they have low signal of on the detector of fluorophore 2. And these cells are not expressing any of the proteins. Now I have like a real two-dimensional histogram. And this is like ideal to see how cells are shown in the two-dimensional histogram. So these cells, here we are studying two different proteins, CD14 and CD4. We are labeling CD14 with PE, and we are labeling CD4 with FACS. We are labeling them with different fluorophores. Then cells in this quadrum are expressing CD14, but they are not expressing CD4, so we have CD4 negative. Cells here are expressing both. They are giving high signal on both of them. So the CD14 positive, CD4 positive. These cells are CD14 and CD4 negative because they are giving low signal on both detectors. And these cells are expressing CD4. They are giving high signal on CD4 detector or on FACS detector, but they are giving low signal on the other detectors, so they are expressing only CD4. This is like a very simple example about how the data looks like in the two-dimensional histogram. Of course, there are many more complex data. So if you want me, I cannot speak about everything in this video. So if you want me to do another video, especially about data analysis and gating, in FACS analysis, please write me in the comments. As you know, now we can in the new FACS machines, we can analyze like tens of proteins and tens of cells in one time, because new FACS machines have like some machines like there is now one FACS machine in the market which contains 10 lasers and 30 detectors. Can you imagine 10 lasers and 30 detectors? So you can analyze many proteins and many cells at one time. But of course, the data then will be much more complex than this data. This is a very simple data. If you want me to talk more about this, then write me in the comments. If you enjoyed the video, don't hesitate to like the video, share, subscribe to the channel. If you have any questions or any suggestions for other topics, write me in the comments, and see you in the next video. Bye-bye.
Okay. Now this fluoro 1, fluorophore 2 with CD4 FITS, CD4 14 PE having different dyes. We can differentiate your cells according to their sizes, according to their morphologies, according to their attachments. So we can have a very nice differentiations of our molecules at the end. So I hope this FACS analysis is pretty much clear today. Yeah, so let's continue further to know from the industrial perspective how the FACS is being used in the industry. Just 2 minutes thing.
Cell processes are controlled by complex and dynamic intracellular signaling networks. Surface receptors pass information into the cell by modifying cytoplasmic proteins. These modifications include phosphorylation, methylation, and acetylation, which control a broad range of cellular functions including growth, development, and apoptosis. Disruption of signaling networks often leads to disease. To understand signaling processes in normal and pathological conditions, identification and quantification of signaling proteins is necessary. Flow cytometry is ideal for such a task. Flow cytometry is a laser-based technology widely used to phenotype cells using antibodies directed against surface markers. It can also allow simultaneous analysis of multiple intracellular targets using antibodies that detect signaling proteins, their post-translational modifications, epigenetic modifications, and downstream transcription factors. The first step of intracellular flow cytometry is fixation of the cells using formaldehyde, which immobilizes proteins and preserves transient signaling events. Cells are then permeabilized with methanol or detergent, allowing antibodies to access to the intracellular space. Optimal fixation and permeabilization conditions depend on the location and biochemistry of the target epitopes and should be carefully considered to ensure successful antigen detection, especially when staining surface and intracellular proteins simultaneously. Since multiple signaling proteins and surface antigens can be identified using fluorescently distinct antibodies, flow cytometry can readily characterize signaling profiles at the single cell level within phenotypically distinct populations. This allows the characterization of the relationship between intracellular targets and enables detection of signaling events from rare cell populations within a heterogeneous sample. Multiplex analysis of these protein targets saves time and resources while producing data-rich results, making intracellular flow cytometry an invaluable tool to unravel the complexities of cell signaling. For more information on validated products and optimized protocols for flow cytometry, visit www.cellsignal.com/flow cytometry.
And the last one,
in this animation, we will introduce you to the flow cytometry experimental technique. Flow cytometry is used to count and analyze the size, shape, and properties of individual cells within a heterogeneous population of cells. Flow cytometry data is extremely quantitative and can be analyzed in depth by specific flow cytometry software programs. Once a suspension of individual cells has been prepared for flow cytometry analysis from cells, tissues, or organisms, the prepared sample is placed on the flow cytometer. How does a flow cytometer analyze the size, shape, and properties of individual cells? A flow cytometer contains several key components, including the sample, fluidics that move the sample into the flow cytometer, lasers, optics which gather the light, detectors to sense the light, and a computer system to output the data into a form that can be analyzed by the researcher. Once a sample has been placed in the flow cytometer, the machine will suck up the sample, mix the sample into a stream of saline solution in the cytometer, and lead the cell suspension through a narrowing channel, causing the cells to form a single file line before the cells pass through the laser at the interrogation point. Each cell within the sample then moves across the laser beam, allowing each cell within the sample to be individually analyzed. As each cell passes through the laser beam will scatter in multiple directions. The flow cytometer detects light scattered in a forward manner, called forward scatter, and light scattered in a sideways manner, called side scatter. The amount of forward scattered light for each cell is detected by a detector on the far side of the cell from the laser. Forward scatter is proportional to the size of the cell. The detector converts the scattered light into a voltage pulse, which is directly proportional to the amount of forward scattered light. The computer converts these data into a histogram plot with the amount of forward scattered light on the x-axis and the number of cells on the y-axis. The amount of side scattered light is detected by a detector located perpendicular to the path of the laser beam. Side scatter is proportional to the shape and internal complexity of a cell. Just like with forward scattered light, the flow cytometer converts the detected side scattered light into a voltage pulse, which is directly proportional to the amount of side scattered light. By analyzing the forward and side scatter data together, the researcher can understand a cell's size, shape, and complexity. In addition, the analysis of the forward and side scatter data can allow the researcher to divide the heterogeneous population of cells into individual populations with varying size, shape, and complexity. This capability of analyzing multiple populations within a sample is the immense strength of the flow cytometry experimental technique. In addition to analyzing a cell's size, shape, and complexity, a flow cytometer can also detect emitted light from excited fluorescent molecules, such as fluorescently labeled antibodies or fluorescent dyes or stains. For the rest of this animation, we will focus on the detection of fluorescent signal by the flow cytometer. For more information about fluorescence, please see the fluorescence animation. The fluorescent molecules, or fluorophores, are excited by the correct wavelength of laser light when the cell passes through the laser beam. After excitation, the light emitted by the fluorophore is directed along a path with emission filters that allow for the detection of multiple different fluorophores emitting light in a cell. A flow cytometer can detect the presence of a fluorophore and also quantify the relative amount of a fluorophore within a cell. A flow cytometer can also be used to detect light emitted from multiple fluorophores in the same sample. While it is common for flow cytometers to detect 2, 3, or 4 colors at the same time, some flow cytometers can even detect up to 18 different colors at the same time. The data can then be presented to the researcher in the form of a histogram or a dot plot. Such data furthers the powers of flow cytometry analysis by enabling the researcher to analyze individual populations of cells within a heterogeneous mix of cells in the sample.
Okay. So that was all about facts analysis for today. And yeah, so today we will not have class in the evening. I will be traveling to Kanpur. So that's why I took this class in the morning. I hope other students will join the recorded version later, the same, and could get benefit out of it. So thank you very much. That's it for today. Tomorrow we will see in the evening or in the morning. So I will let you know about how the internet conditions are there. So according to that we will meet. We might not see us on the Google or on this YouTube maybe because of the streaming problem, but maybe on the Google Meet then. Okay then, take care. Bye bye.